shoot formation. So formation of auxins and cytokinins in various ratios. So that leads to the formation of organogenesis, such as namely rhizogenesis and coalogenesis. Rhizogenesis. So it means development of roots by the adding of high amount of auxins and less amount of cytokinins. So that leads to be formation of roots. So by a process that is called rhizogenesis. And similarly, adding a high amount of cytokinins and less amount of auxins, that leads to be the formation of shoot. So by a process that is called coalogenesis. So rhizogenesis and coalogenesis. So both are formed by the different percentages of auxins and cytokinins. So in the year of 1957, Skoog and Miller demonstrated the role of auxins and cytokinins on the root and shoot formation in tobacco. Later in the year of 1962, the medium, culture medium, which is prepared by the scientist Murashige and Skoog. Murashige and Skoog. Murashige and Skoog. So these two were scientists developed a culture medium. So that is namely Murashige Skoog medium or MS medium for using of tobacco culture. And in the year of 1987, by using of tissue culture, first isolation Bacillus thuringiensis genes from bacterium Bacillus thuringiensis. So first gene which is formed BT gene. So BT gene extracted from the bacteria, that is namely Bacillus thuringiensis. So this is the brief history of tissue culture. Next you see, so in the year of 1902, Herbert Land attempted to culture isolated mesophil cells, but not succeeded. Culture isolated mesophil cells. So mesophil cells which are mainly present in the leaves. So mesophil cells which are nothing but chloroplast containing cells, they are called mesophil cells. So in the year of 1902, Herbert Land not succeeded by using of mesophil cells. In the year of 1937, R.J. Gathret first prepared a callus culture of carrot. In 1957, Miller and Skoog put forth the hormone hypothesis. Later, Guha and Maheshwari in the year of 1966, Guha and Maheshwari cultured pollens, obtained haploid plants. So fundamental principles. So in that fundamental principles, one is the totipotency and the second one plasticity. Totipotency. So the cell which is ability to divide and formation of new plants, means entire plant cell divide and produce new plant. So that capable capacity to the cell divide and produce more number of cells, and these cells again and again divides and produce number of cells and tissues, and both are associated and forming completely a new plantlet. So that is called totipotency. So once you see the totipotency, so this is totipotency. You see, so this is a cell which is extracted from a plant, and these cell divides and produce more number of cells. Then next, so these all the cells which are together fused and forming a organ, and number of organs which are formed later, these are associated and developing a complete plant. So by this process, this is called totipotency. Then next, అరే అల్లరి చేయకుండా అమ్మా, ఏమంది కబ్బా. So next you see, one is the totipotency and second one plasticity. So in plasticity, so the ability of plants to alter their metabolism So in plant tissue culture, there are totipotency and plant plasticity. So plant tissue culture depends upon, one is the totipotency and the second one plant tissue plasticity. Plasticity, it means ability of plants to alter their metabolism, growth, and development to best suit their environment. That is called plasticity. The next, explant. So explant, a part of tissue which is extracted from various parts of the plant. Explant to be extracted from a plant in various parts. So plant tissue cultures generally initiated from multicellular tissue fragments. That is called explant. So these explant obtained from living plants. So in that living plants, the explant taken from leaf, stem, root, petiole. So petiole, it means stalk of leaf, petiole. Hypocotyle. So it is a seed coat. Then next, cotyledon, embryo or meristems. So these parts which are act as a explant. Then next, selection of explant. You see in this image, this is a plant, explant taken from seeds or from leaves. The explant is selected either haploid or diploid. Then next, there are given the plant growth can be achieved in two ways. Shoot directly by appropriate media, by somatic embryogenesis. Next, sterilization. Sterilization. So plant tissue culture, it is a method So these are the sterilization of the plant material. Explant could be sterilized. Explant could be sterilized by using of different chemicals. One is the liquid detergent and ethyl alcohol. Then next to sodium hypochlorite. Next, culture media. Now these explants placed on the surface of the culture medium, but sometimes these are directly placed into a liquid medium. So liquid media, basal media, solid culture media or liquid media. So the main difference between the liquid media and culture media, by adding of agar agar, the culture media that is solidified, so that is called solid culture medium. If do not adding agar agar, now the distilled water with macronutrients, vitamins and minerals and amino acids, so that is called liquid media. By adding of agar agar, that is culture, solid culture medium. Do not adding agar agar, that is a liquid media. In the culture media, that is composed of inorganic salts, organic nutrients, vitamins, and plant hormones. So plant hormones, that is auxins, cytokinins, and a few amount of gibberellins. So that is preparation of culture media. Next, so there is culture media can grow and forming a number of cells, that is called callus. So you see, this is composition of nutrient medium. So different type of nutrients and their composition, micronutrients and macronutrients. Micronutrients which are added to the culture media, that is namely nitrogen, phosphorus, potassium, calcium, magnesium and sulfur. Macronutrients, that they are borons, chlorine, cobalt, copper, iodine, iron, manganese, molybdenum, sodium and zinc. So you see, next, the salts which are used, nitrogen, magnesium, calcium, potassium. The next micronutrients, copper, nickel, molybdenum and cobalt. Vitamins, amino acids. So in amino acids, mostly we are used, that is glycine. In carbohydrates, sucrose. So these are different cultured mediums prepared by different scientists. One is the White's medium, MS medium, B5 medium, and N6 medium. So in these medias, most of the cases we are used as only MS media. So MS media, so Murashige and Skoog. So these media that is proposed by two scientists, one is the Murashige and Skoog. So it is widely used for many types of cultured systems. So you see, so this is the steps in the plant tissue culture. First, selection of parent. Next, isolation of parent. Next, sterilization of explant. Next, inoculation. Next, incubation. Initiation of callus. Subculturing. Regeneration. Hardening. Next, transfer of plantlets to greenhouse or open field. So these are different types of plant tissue cultures: seed culture, meristem culture, protoplast culture, organ culture, pollen culture, callus culture, cell culture, and embryo culture. So there are different types plant tissue culture types. Now you see, so these are plant tissue culture applications. So by using of plant tissue culture, the commercial production of plants used as potting, landscape, and florist subjects. So plant tissue culture has several applications in the fields of agriculture, horticulture, forestry, pharmacology, medicine, and environment. So first one, so by using of plant tissue culture, it is possible to produce a large number of plants within a short time and space through the tissue culture. And a mass production of plants through the tissue culture, that is known as micropropagation. So production of high proportion of plants produced by a short time, that is called micropropagation. So this micropropagation technique is applied for propagation of ornamental plants, orchids, and other exotic plants, fruits, and plantation crops. The next, plants regenerated through tissue culture. So that shows variations. That is called soma-clonal variations. By using of plant tissue culture, so it should be show some variations, and that variations are called soma-clonal variations. So these soma-clonal variations are used for crop improvement. Next, in some plants like papaya, it is a unisexual plants. So male and female plants are separated. So in that, female plants are needed for greater propagation to produce more fruits per cultivated area. So it means these female plants are selectively produced through the tissue culture. And viral diseases from vegetatively propagated plants, so they can prevented by producing virus-free plants from shoot tip culture. So, and the using of tissue culture, your formation of artificial seeds. So artificial seeds or synthetic seeds. So these artificial or synthetic seeds produced from somatic embryos by encapsulation with sodium alginate. And these somatic embryos can be easily stored, germinated, and transported. The tissue culture of medicinal plants helps in the in vitro production of high value products. So these high value products which are used for industrial and medicinal importance. And the production of transgenic plants by transfer of foreign genes, and that should be completed by dependent on the plant tissue culture. Dependent on the plant tissue culture. So these are the different types of tissue culture applications, and some are given. So in this plant tissue culture applications, in that commercial production of plants used as potting, landscape, and florist subjects, and conserve rare or endangered plant species. By using of plant tissue culture, we can cultivate more number of plants endangered. So these endangered plant species, in that taken an explant from these endangered species, and these explant using by in vitro tissue culture and producing more number of plants by a short time. Next, to screen cells rather than plants for advantageous characters, herbicide resistant and tolerance. Next, large scale growth of plant cells. So large scale growth of plant cells in a liquid culture in bioreactors for production of valuable compounds. That is secondary metabolites, recombinant proteins, and biopharmaceuticals. So you see this chart. So in this image, so these are all the advantages and applications of plant tissue culture. In that, clonal propagation, large scale multiplication, biomass energy, breaking dormancy, secondary metabolites, wide hybridization, generic variability, disease-free plants, haploid, polyploids, and triploids, international exchange of germplasm, somatic hybrids or hybrids, fast multiplication, synthetic seeds, early flowering, generic transformation, somatic embryogenesis, overcoming self-sterility, cryopreservation of germplasm. So these are the different advantages of tissue culture. So in that, in our syllabus, they are given some of the applications. In that first one, clonal propagation, micro propagation. Clonal propagation. Propagation, it means production. Clone, clonal, clone. So clone, it is similar to the parent cell. So it means it is a clone. It means it is a Xerox copy of the parent. So in that plant population that is derived from a single donor plant. So that is called as a clone. And the multiplication, genetically identical copies of the cultivator, that is called clonal propagation. So this clonal propagation may be useful tool to get a large population of plant species having desirable traits. So desirable traits, it means good quality characters. We can take as a good quality characters. So in this tissue culture from a callus, So callus is nothing but group of cells. So explant undergoes two repeated divisions and forming a group of cells. So that is named as callus. So in this tissue culture from a callus, large number of shoot meristems can be regenerated within a very short time and a short space. So in this tissue culture technique, callus undergoes to repeated divisions and forming a high number of cells or organs by a short time and a short space, a less space. So as a result, number of plantlets, it means more number of plantlets can be produced from a short time and short place by using of callus in tissue culture. The next soma clonal variations. Soma clonal variations or genetic variability. So these soma clonal variations. So variation generated by the use of tissue culture cycle has been termed as soma clonal variations. So soma clonal variations. So it means so explant, it is a vegetative part. Now this explant cells undergoes to repeated divisions and forming culture. So in this culture having number of cells, so in that cell, some of the cells shows variations. So that variations is called soma clonal variations. So these soma clonal variations first identified by the scientist Larkin and Skewsoft. So this genetic variability is due to cells of various ploidy levels and the genetic constitution of the initial explant. So these kind of variations may show some useful characters such as resistant to the particular disease. Some are shows herbicide resistance and stress tolerance. Next, secondary metabolites. Production of secondary metabolites. By using of tissue culture, we can produce more number of secondary metabolites such as alkaloids, steroids, glycosidic compounds, and many essential oils and flowering and coloring agents by using of plant tissue culture. Alkaloids, namely vincristine and quinine. Steroids, diosgenin. Glycosidic compound, that is digoxin. Essential oils from jasmine. Coloring agents from saffron. So by the production of secondary metabolites, using of tissue culture technique. Digoxin, it is a secondary metabolite. It is extracted from the plant that is Digitalis lanata. So these digoxin uses in cardiovascular disorders. And another one, diosgenin. Diosgenin, it is also one type of secondary metabolite. So these diosgenin extracted from the plant that is Dioscorea deltoidea. So these diosgenin used as anti-fertility. Next, saffron. Saffron extracted from the plant that is Crocus sativus, and these saffron used in food industry for coloring agent. From the time of immemorial, man has been dependent on the plant products besides the supply of food from plants. So these plant products, mostly secondary metabolites, include pharmaceuticals, flavors, perfumes, agrochemicals, insecticides, and raw materials for industries. So chemically, the plant products may be alkaloids, terpenoids, or glycosides. So steroids and phenolics are glycosides. So according to World Health Organization survey, nearly 70 to 80% of the world population depends on herbal drugs. So it is a fact that many chemicals with chemical structures that cannot be chemically synthesized can be conventionally produced and produced in plants. Chiconine is a dye produced by the cells of lithospermum on a commercial scale. So these secondary metabolites production through the tissue culture. So the one of the important new technique for large scale production of secondary metabolites. So that is a tissue culture technique. So in this technique, first selection of lines for high yield of secondary metabolites. And in this technique, cloning, cell aggregate cloning. So these are used selected cells in a cultured media, that is on agar medium. And these are gives calls for large scale production of cultured bioreactors. Certain secondary metabolites emit fluorescence under the ultraviolet, and the corresponding clones can be identified. Some workers use simple sensitive and analytical methods for quantitative estimation of desired metabolites. And certain cells resistant to toxic compounds. So it maybe leads to the formation of mutant cells, which can ever overproduce a primary metabolite. And the next one, breaking of seed dormancy. Seed dormancy. So if the all conditions would be applied to the plants, some of the seeds do not germinate and produce new plantlets. So that is called seed dormancy. So by using of tissue culture technique, so seed dormancy should be breaking by using of tissue culture technique. So in that employing embryo culture, the seed dormancy period can be reduced or eliminated. Now the second one, the breeding cycle can be shortened in many of the plants. The life cycle of iris was reduced from 2 to 3 years to less than 1 year. So these are the main applications using of tissue culture. So once again, I repeated. So the main applications are, one is the clonal propagation, large scale of multiplication, biomass energy, breaking of seed dormancy, second, production of secondary metabolites, and hybridization, genetic variability, diseased-free plants, haploids, polyploids, and triploids, international exchange of germplasm, somatic hybrids or hybrids, fast multiplication, synthetic seeds, early flowering, genetic transformation, somatic embryogenesis, and overcome self-sterility, cryopreservation of germplasm. So in that soma clonal variations, so it means the genetic variations that are found in the in vitro cultured cells. Some of the genetic variations are found in the cultured cells. So these genetic variated cells collectively called as soma clonal variations. The plants derived from such cells are referred to the soma clones. The next, Cali clones and protoclones are represents cultures obtained from callus and protoplasts respectively. The growth of plant cells in vitro is an asexual process involving only mitotic divisions of cells. So these soma clonal variations, the term first used by Larkin and Scowcroft in the year of 1981. For variations arising due to culture of cells, variability generated by a tissue culture. So the soma clonal variations which are formed in cultured plant cells by mainly two reasons. One is the gene related variations and the second one mutation related variations. So these are the advantages of tissue culture.